Hello, thank you for your interest in the poster Effect of Small Mobile Stem Cells on Alveolar type 2 lung cells at gene expression level. My name is Abdulkader Ramo and I am at SMSbiotech company. Alveolar type 2 or AT2 cells are main components of human lung alveoli and essential progenitors for its regeneration. Small mobile stem cells are isolated from human peripheral blood and cultured in vitro extensively. These have many important features such as resilience, extracellular matrix production, high capacity for proliferation, and angiogenic properties. SMS cells can stimulate the proliferation of human alveolar type 2 cells in the absence of added serum to the media. This is mimicking lung injury conditions with damaged blood vessels. Furthermore, SMS cells have a significant effect on the gene expression of alveolar type 2 cells. Within 3 days of co-culture, we have detected gene expression changes at more than 1,300 genes. The changes in gene expression relate to lowering inflammation, reducing apoptosis, pathogen protection, angiogenesis, and other features which are relevant to cell therapy of chronic obstructive pulmonary disease emphysema. SMS cells may overcome many of the limitations of adult-derived cells that are currently being tested for COPD cell therapy. These include limited availability, donor variability, in vivo cell death, and exclusion to direct administration to the lung. The strong binding of SMS cells to alveolar type 2 cells and the clear effect exerted at gene expression level will hopefully lead to a mechanistic approach for lung cell therapy. SMS cells' functional features establish actually the paradigm of the interaction between a stem cell and an unrelated progenitor cell. All that in the potential context of a multimodal cell-induced therapy. Thank you very much for your attention.